Hello student. After a long gap, means almost after 6 month gap, once again welcome to honors classes. Today I am going to upload one previous year biology questions paper for your upcoming class 12 final examination. From now I am going to serially upload the previous year last 10 years question paper for you for your coming exam. Means question papers from 2012 to 22 with solution I am going to upload serially, and after that I am going to upload the important questions from each chapter of biology, and just before your exam I am going to upload the most probable questions which are important for your upcoming final examination. So let's proceed to the video. Today I am going to upload the question paper 2016 biology, and first part, it contained a botany part of 35 marks. So first question is, why grafting is not possible in monocot? So you know that means in monocot it is not possible, since in monocot the cambium tissue is absent. So cambium tissue absence means, you know that cambium is a meristematic tissue. They have the power of division. Since this meristematic tissue is absent in monocot, that's why grafting is not possible in monocot. This question is carrying 1 marks. Name unicellular fungi which reproduces asexually by budding. Example is yeast. This is the questions from first chapter. What were the raw material used in Miller experiment? This is from genetics. So the experiment used are water, H2O, methane, CH4, ammonia, NH3, and hydrogen, H2. What is hidden hunger? So hidden hunger or micronutrient deficiency. So if in question paper the micronutrient deficiency term also come, you don't confuse. It means it occur when the quality of food that people eat does not meet their nutrient requirements, so they are not getting the essential vitamins and minerals they needed for their growth and development, is called hidden hunger. Next question, which is carrying 2 marks. Define antigen and antibody. An antigen is a foreign substance that enter your body, and this can include bacteria, virus, fungi, allergens, venom, and other various toxins. An antibody is a protein produced by your immune system to attack and fight off these antigens. Antigens means these are foreign particles, and antibody are produced in our body. How DNA is isolated in purified form of a bacterial cell. DNA, a genetic material, is isolated in purified form by treating the bacterial cell with the enzyme. The name of the enzyme is important, that is lysosome, and remove the cell wall. Draw the LS of pistil of angiosperm, which is ready to take part in the process of fertilization. So it means you have to draw the diagram of female gametophyte. If in place of this question, draw the diagram of measure of female gametophyte also come, then also you have to draw this diagram. And here, these are the pollen grain. So now this is the female pistil, mature pistil structure, which is ready for fertilization, and within the pistil there are synergies are present, egg cells are present, antipodal cells are present, and at the same time polar nucleus are also present. Next, what are the cells present in a developed embryo sac of the angiosperm? So what are the cells? There are total 7 cells are present. The 3 are antipodal cells. These 3 form the egg apparatus. In the egg apparatus, the central cell is known as egg cells. Outer 2 cells is known as synergies. And in the center, there is a cell which has 2 nuclei. Name is polar nucleus. So this is also known as 7 cell 8 nucleate stage. So if question come, write the 7 cell 8 nucleate stage, then also you have to draw the same diagram. What is polyploidy? Name the chemical used for induction of polyploidy in plant. Polyploidy is the heritable conditions of possessing more than 2 complete set of chromosomes. Means when more than 2 set of chromosome is present, then it is called polyploidy. And this is the term which is very important, COLCHICINE. So you remember the spelling is widely used mitotic inhibitor for the induction of polyploidy in the plants during the cell division by inhibiting the chromosome segregation, because you know that in cell division the chromosomes are segregated equally, but this chemical is used which means stop the which prevent the chromosome to segregate equally. Next question, what are the interspecific hybridization? The cross between animals and the same genus but different species is known as interspecific hybridization. For example, liger is an example, which is an offspring of female tiger and male lion. Name two antibiotics that are produced from the bacteria along with the name of the concerned bacteria. These are the name of the antibiotics. Suppose streptomycin are produced from streptomycin, and bacillus, which is produced from Bacillus subtilis. Name of the microbes used for production of acetic acid and lactic acid. So, acetic acid is produced by Acetobacter acetobacterium, and lactic acid is produced by Streptococcus lactis bacterium. Next, how light influences the growth and development of plant. Light intensity influences the manufacture of plant food, stem length, leaf color, and flowering. Generally speaking, plant grown in low light tend to be spiny with light green leaves. A similar plant grown is very bright light tend to be shorter and better branches and have larger dark and green leaves. Or what are the special characteristic of parasitic plant? So these are the special characteristic of parasitic plant. They have special kind of root called the hostelria, which penetrate into the host tissue and have contact with the vascular tissue to draw the water and food or both from the host plant. If they are total parasite, then they don't have leaf to carry out the photosynthesis. If they are partial stem parasite, they may have leaves, but they only absorb water from the host plant. In the parasitic sorry mode of life, the parasites gets benefited whereas the host is affected. In other words, the parasite live at the cost of the host food. Next question, 2 marks. Write notes on any two food chain. For food chain, you get in EVS of your first year also. So food chains may be defined as the transfer of energy and nutrients from the source or in plants through a series of organisms with repeated process of eating and being eaten. For example, Mars vegetation is eaten by grasshopper, then grasshopper is consumed by shrew, and then shrew is consumed by auk or owl. And this is the drawing of food chain. Then next is trophic level. Trophic level stays in a nutritive series of food chain of an ecosystem. The organisms of chains are classified into this level on the basis of their feeding behavior. Means each level or each organisms is grouped as different trophic level. The first and the lowest level contain the producer, green plant. The plant and their product are consumed by the second level organism, the herbivores or the plant eater. At the third level, primary carnivores or the meat eaters eat the herbivore, and the last fourth level, secondary carnivore eat the primary carnivores. So these are the different trophic levels. Means grass is the first trophic level, then it is eaten by rabbit. Rabbit is the second. A rabbit is eaten by snake, so it is the third. Next, eagle is the fourth trophic level. Write the scientific name and the utility of the following plant. So these three plants are given. First one is Cinchona. So this is the scientific name of Cinchona, Cinchona officinalis Linn, and they are used specially as a treatment for fever and malaria. Next is neem. This is the scientific name of neem, and this is the medicinal use of neem. Means neem is used as to blood control the blood sugar level, heal the ulcer of the digestive tract, etc. Next is Rauwolfia. Scientific name is Rauwolfia serpentina, and they are used to treat the high BP. Next, or another question is orwood. They write the scientific name and their families of three important timber yielding plant of Assam. So here I am giving the name of three timber yielding plant and with their family. So, with family is also given. Next, explain the significance of mutation in evolution. A sudden mutation means what you know. A sudden change in genetic material is known as mutation. Mutation plays an important role in evolution. The ultimate source of all genetic variation is mutation. Mutation is important as the first step of evolution because it creates a new DNA sequence for the particular gene and create a new allele. Since genetic change takes place, that's why new allele or new offspring will be formed. A classic example of mutation in the evolution of humans is the mutation in the gene HBS, which makes the RBC. One copy of this gene provides the resistance to malaria, but two copies of this gene causes the sickle cell anemia. Or discuss the modern synthetic theory of evolution. The modern synthetic theory of evolution is also known as modern synthesis. So if this term is given in question paper, don't confuse. Merge the concept of Darwinism evolution with Mendelian genetics, resulting in unified theory of evolution. This theory is also referred as New Darwinism. So if New Darwinism term also come, then you have to write the same theory. It describes the evolution of life in terms of genetic changes occurring in the population that leads to the formation of new species. It also describes the genetic population or Mendelian population, gene pool and gene frequency. The major concepts coming under this theory include the genetic variations, reproductive and geographical isolation, and natural selection. Next question, discuss the major steps involved in the gene cloning. So gene cloning involves the separation of specific gene or DNA fragments from a donor cell. Means specific or required DNA is isolated from the donor cell, attaching to a small carrier molecule called a vector, and then replicating this recombinant vector into a host cell so that we can get the desired character. Step involved in the gene cloning is isolation of donor DNA fragment or gene. Second step is selection of the suitable vector from means where the gene means desired DNA is input. Incorporation of the donor DNA fragment into the vector, transformation of the recombinant vector into a suitable host, and isolation of the recombinant host cell. And this is the diagram of the, I am giving the diagram of the gene cloning also. Look, this is the E. coli bacteria. Here the plasmid is there. So first plasmid is isolated is the first step. Second step, cut the plasmid with the enzyme, means with the molecular scissor the plasmid is cut here. And then in another side, this is the cell with DNA which gene of interest means required. The gene which we need is present in this DNA. Then first this DNA is isolated. Then the DNA is segmented into different parts with the help of molecular scissor and cut the cells of DNA with the same enzyme. And then this red portion is the gene of interest. This portion is separated and this portion is combined with the targeted, this means plasmid. And then with the help of DNA ligase enzyme, they are jointed with this plasmid. And as a result, the recombinant DNA plasmid is produced or present, means where the gene of interest is present. Then this plasmid is put into the bacterium by the transformation process and then allow the bacteria to reproduce. And finally, lots of bacterial cell will be produced where the gene of interest or required gene will be present. So this is the process of gene cloning. Or the state the basic steps involved in the genetical modification of an organism. So genetical modification of organism means another term, you can say it as genetic engineering also. So genetic engineering or genetical modification of organism is the same thing. So is accomplished in the basis three basic steps. These are isolation of the DNA fragment from a donor organism. Second, the insertion of the isolated donor DNA fragment into the vector genome. And third, the growth of the recombinant vector in an appropriate host. And this, the diagram, in diagram, everything is given here. So first step is the plasmid is removed. Then again the plasmid, second step, the foreign DNA is cut by the same enzyme and the foreign DNA is inserted into the tDNA in the plasmid. Then the plasmid is again reinserted in the bacterium. Then it undergo replication and the plant cell is grow in the culture, and finally lots of desired plant offspring or seedling will get. Next step, why is the pond regarded as a good example of ecosystem? Explain the path of energy flow in the pond ecosystem. So the abiotic component are water, air and sunlight. The fishes survive on the oxygen dissolved in water. They feed upon the aquatic plant and microbes, consume all the dead matter, cleaning the water all by itself. Thus a pond is self-sustaining ecosystem, means everything are present in the pond itself, water of the pond itself. That's why it is counted as a good ecosystem. Pond get their energy from the sun. As with other ecosystem, plants are the primary producer. The chlorophyll in the aquatic plant capture the energy from the sun to convert the carbon dioxide and water to the organic compound and oxygen through the process of photosynthesis. Next question, discuss the process of development of a tree cell pollen grain from a microspore mother cell with the help of a label diagram. So the process, sorry, this should be the spelling mistake. He has come. This should be the the process of formation of microspore from a pollen mother cell is called microsporogenesis. Microspore, as they are formed, arranged in a cluster of 4 cells of microspore tetrad first. Then the anthers mature and dehydrated and the microspore dissociated from each other and develop into pollen grain. From each microspore tetrad, each cell develop into a pollen grain. When the pollen grain is matured, it contains two cells. First one is vegetative cell, second one is generative cell. The vegetative cell is bigger, has abundant food reserve and a large irregular nucleus. The generative cell is small and floats on the cytoplasm of the vegetative cell. It's spindle-shaped and dense cytoplasm and a nucleus. In over 60% of the angiosperm, pollen is set at the two cell stage. In the remaining species, generative cell divide mitotically to give rise to two male gametes before pollen grains are shed three cell stage. So, first, this is the microspore mother cell or pollen mother cell. First, it undergo meiosis division and two cells are divided. These are the microspore dyad. Then again they undergo division and finally four microspore tetrad is formed. From four microspore tetrad, is from microspore is produced. This is the microspore. This microspore has two nucleus. This is the nucleus, and this nucleus is divided. One is vegetative cell, another is generative cells, and this generative cell is in spindle shape. So this is the structure of the tree cell structure development of pollen grain. Next question. What is ecological pyramid? Explain different types of ecological pyramids. An ecological pyramid is a graphical representation designed to show the biomass or bioproductivity at each trophic level in a given ecosystem. And there are three types of ecological pyramid. In class 11, you have got these are pyramid in number. The second one is pyramid in biomass. And third one is pyramid in productivity. Sorry. Next, this botany section is finished. Now go for the zoology section of the same question paper. So zoology section also containing 35 marks. So first question, fill in the blanks. So any two you have to write, but now you have to prepare all the question because you don't know which question is going to be coming in your upcoming exam. So first, silkworm belongs to the order. This is the Lepidoptera. Next, the milk of the rosy, the first transgenic cow, contained the human alpha, and this is the lactalbumin. Next, the structure which provide the connection between the developing fetus and the uterus of the mother is called what? This answer is placenta. Acrosome of the sperm is formed from where? The Golgi apparatus. Next question, answer I need to express the genotype of AB blood group. How many genotype will be present? There are total 6 different genotype will be present there. What is the algal bloom? An algal bloom is a rapid increase or accumulation in the population of algae in the freshwater or marine water ecosystem. Write the equation of species area relationship in a logarithm scale. The species area relationship is expressed by the equation log S is equal to log C plus Z log A. And this is the diagram. Next, write the scientific name of the water hyacinth, that is Eichhornia. This is important for your viva of your practical exam also. Next, write answer of any four of the following, that is explain the chromosomal theory of inheritance. The chromosomal theory of inheritance was given by Boveri and Sutton in the early 90s. It is the fundamental theory of genetics. According to this theory, genes are the unit of heredity and are found in chromosomes. Then what is Leydig cell and state its function? These are the interstitial cells located within the seminiferous in the testis and adjacent to the seminiferous tubules in the testis. The best established function of Leydig cell is to produce the androgen testosterone under the control of luteinizing hormone. Next, write notes on sickle cell anemia. Sickle cell anemia is one of the group inheritance disorder known as sickle cell disease. It affects the shape of the red blood cell which carry the oxygen to all part of body. Blood cells are usually round and flexible, so they move easily through the blood vessels. In sickle cell anemia, some blood cells are shaped like sickle or crescent moon. These sickle cells also become rigid and sticky, which can show and block the blood flow. Infections, pain, fatigue are the symptoms of the sickle cell disease, and treatment include the medication, blood transfusions, and rarely a bone marrow transplant are the treatment. Step 4, features of genetic material. It must be stable. It must be capable of being expressed when needed. It must be capable of accurate replication. It must be transmitted from parent to progeny without sin. What are the barrier of innate immunity? So these are the barrier. Innate immunity means the immunity which is inborn and inherent mechanism that an organism is born with, and there are lots of barrier. Some of these are anatomical or chemical barrier. Second one is phagocytic barrier. Then third is plasma protein barrier. This is the plasma protein barrier. And fourth one is cytokinesis. Next, state the difference between any two. First one is sex chromosome and autosome. You know, this is a difference. So here only 2 marks is there. I am giving you 4 difference, but if 2 marks come, you just write 2 difference. 2 difference will be sufficient. Next difference will be T lymphocyte and B lymphocyte. So here also I am giving you for 3 difference. So if 2 marks come, you just write 2 marks. Next, grazing food chain and detritus food chain. These are the difference of grazing food chain and detritus food chains. Then follicular phase and luteal phase, and these are the 2 phases of menstrual cycle. Both are important phases. One is follicular phase and one is luteal phase. For better explanation, you can go to my another video of human reproduction. There I will explain briefly about the follicular phase and the luteal phase. Draw the label. The internal structure of the seminiferous tubules. So seminiferous tubules, these are the basement membrane. Next is the primary spermatocytes, secondary spermatocytes. These are the Sertoli cells which give nutrition to the upgrowing sperm. Then these are the sperm and these are the spermatid. Next is write briefly on prevention of AIDS. So AIDS means refers to the acquired immunity deficiency syndrome causes by HIV virus, and there are some preventive measure of AIDS are using one-use needle, practicing protected sex, that is use of safety like condom, regular health checkup, and regulating blood transfusion and pregnancy can help in the prevention of AIDS. One more imperative influence is the awareness of AIDS. Next, what do you understand by fishery? State its importance. So fishery refers to this is the definition of fishery, and what is the why fisheries means important. And in the next para I am giving about this. Fishery refers to the enterprises of the raising or harvesting fish and other aquatic life, and also the site where such an enterprises is set up. This is the definition of fishery, and these are the importance of fishery. Next, what are the consequences of loss of biodiversity? The biodiversity loss can have significant direct human health impact if ecosystem services are no longer adequate to meet the social need. Indirectly, changes in ecosystem service affect the livelihood, income, local migration, and on occasions may even causes or exacerbate political conflict. Write the Mendelian law of inheritance. So here, the first law of Mendelian is told about is the segregation state that every individual possesses 2 alleles and only one allele is passed on to the offspring. The law of independent assortment means second law state that the inheritance of one pair of gene is independent of inheritance of the another pair. Next, explain the incomplete dominance. Incomplete, sorry, here should be the I. So incomplete dominance result from the cross in which each parental contribution is genetically unique and give rise to progeny whose phenotype is intermediate. Means suppose this is the father is red and mother is white, then the offspring of progeny will be pink. It is neither same with it as it is with its father nor its mother. Means it has an intermediate color. How a healthy person acquire pneumonia and what are the causative organism, and state the symptoms of this disease. So how pneumonia can causes? So these are how a pneumonia can causes. Means it is a lungs infection causes by bacteria, virus or fungi, and main causal organism is Streptococcus pneumoniae. And symptoms of pneumonia are first one is cough, second one is difficulty breathing, third one is rapid heartbeat, fourth one is high temperature, feeling generally unwell, and sweating and shivering. Next, last question, discuss the methodology involved in the Human Genome Project. The Human Genome Project was an international scientific research project that make use of 2 methodologies that aim at identifying cDNAs and the location of the coding as well as non-coding region of gene, followed by assigning the function of those genes. So these are the methodology I am given here. First one is express sequence tag and second one is sequence annotation. Next, or another or is given there, that is the discuss the process of translation of genetic code. The genetic code is a set of three-letter combination of nucleotide called the codons, each of which correspond with a specific amino acid or stop signal. Translation occur in a structure called the ribosome, which is a factory for synthesis of protein. The process of transpiration, it has several steps, and these steps are: first one is initiation step, here given. Second one is given as elongation step, and third one is given as translation step, and last one is given as termination step. And here I am giving you two diagrams. First one is whole process of translation, and second one is the just the process of elongation. So in exam, if you have sufficient time, then you can draw this diagram, but if there is not sufficient time, you then you can draw this diagram also. But with this answer, you should definitely draw one diagram. Okay, then these are the questions for the 2016. In my next video, I am going to upload another uh, question paper of previous year. Okay, then thank you.